We will start our exploration of diversity of animals with the simplest of the organisms, the poriferans. They are actually the pore-bearing animals, and commonly they are known as sponges. Poriferans or sponges are acoelomate. It means that they lack coelom, which we discussed earlier, and they belong to parazones, which means that they lack proper symmetry and they lack tissue organization. Although there are organization at the cellular level, and this tissue organization increases as we move from simplest animals to the higher animals. About their general characteristics, they live in freshwater forms, and they are also a habitant of a marine environment, and they range in size from few millimeter to 1 meter thick. And their larval stages are moving or movable, while their adult stages are sessile and they remain attached to substratum. If we look at the body plan of poriferans, we can see that there is a single body cavity which is known as spongiocele. It is shown here, and it may be divided into flagellated chambers or canals lined by flagellated choanocytes. These are choanocytes. And then we can see that numerous pores are present in the body wall. The pores through which water enters the body are called as ostia. These pores are called as ostia, while the single pore through which the water or food material moves out of the body is known as osculum. So, and there is no respiratory or circulatory system in poriferans. Since sponges are sessile, therefore these depend upon the food coming to them along with water currents and the movement of flagella of these choanocytes which are present here. And we can see that when the food comes to them, the food enters the spongiocele cavity through the ostia, these pores, and then the food enters the choanocytes, and it is digested, and the waste products either diffuse out of the sponge directly through the body wall or flows out through this osculum. Now, looking at the sponge anatomy, we can see that if we take a single section of this area, we can see that the outer layer of a sponge consists of epithelial cells, and these are flattened surface cells, and these cells are similar to the epidermal cells of higher animals. And next to this layer is a layer of gel-like substance which is known as mesoglea, and inside the mesoglea we can see that there are amoeboid cells, and there are glass-like structural elements which are known as spicules. So, the amoebocytes are multifunctional. They transport and store food, and the spicules are the cells types which are skeletal, or they function as a skeleton. And in addition, we can see that there are choanocytes, and if we enlarge this choanocyte, we can see that it is exactly like type of unicellular protist which is called as Queneau Flagellate we studied earlier, and it seems certain that these protists are the ancestor of the sponges, these Queneau Flagellates. And we can notice here that flagella is present here, and the beating of flagella of many choanocytes that line the body interior draws water in through the pores, through these pores, and thus the sponges bring in food and oxygen and expel waste. Sponges reproduce both asexually as well as sexually. Asexually, they reproduce by budding, or the which are outgrowths on the outer surface of the body, while they can also reproduce asexually by the formation of gemmules, which are buds which are formed on the inner side of the body. We can see here the formation of the gemmule, and these are actually inactive cells. They intend to survive long period of time during unfavorable conditions, and during favorable conditions, gemmule leave the capsule and it develops into a new organism as shown here. And during the sexual reproduction, sperms are released into water and they can travel to the other sponges. And fertilization is internal and take place in mesohyle, and fertilized eggs develop into zygotes in this jelly-like substance, which is mesohyle, and then a zygote become free swimming larvae, and the larvae swim from the body of the sponge or on currents created by the collar cells, which are known as quenocytes, and the larva eventually settle on a surface and it develops into adult that can reproduce again, and most sponges are mobile only in their larval stages. We can see here the sperms are being released and they are taken to the female body, and from there internal fertilization take place and the free swimming larva is produced, which then forms young sponge. The examples of Phylum Porifera are cycone, which is a typical marine sponge, and the other example is that of leucoselenia, and these are tubular sponges. The next example is that of Euplectilla. It is beautiful and delicate sponge, and it is made up of glassy framework, as we can see, and it is sometimes known as Venus flower basket glass. The next example is that of Spongilla, and Spongilla is a freshwater sponge, and these sponges are economically very important, and they are used since ages for washing and bathing, and they are also used in the absorption of fluids and blood during surgical operations, and they are also used in sound absorption in building for making soundproof buildings.